The Genuine Article. Discussion and commentary based on articles from JACC.
Hello, I'm Tony DeMaria, the editor of JACC, here with another edition of Inside JACC. Now, the story about sildenafil and its role in heart disease in general and congestive heart failure in particular is really interesting. We've got a paper in press that deals with the basic science validation of the usefulness of sildenafil potentially in the heart failure setting. And here to discuss that with us is David Kass, well known to all of you, I'm sure. David is professor of medicine and bioengineering at Johns Hopkins. So David, tell us a little bit about the sildenafil story.
So as some of your listeners may know, sildenafil inhibits an enzyme. It's one of the phosphodiesterases, PDE5. This is the first PDE discovered that chews up cyclic GMP, and it was known to be in platelets, and it was known to be in blood vessels, and so when sildenafil was developed as a drug, it was first for an ischemic indication, coronary ischemia, and the trial done in the early 90s by Pfizer in ischemia gave rise to what has got to be one of the most famous side effects of all drug development history, and it really changed the entire course of the development of the drug. It didn't work so well for ischemia. Worked great for this other indication, and erectile dysfunction was born. And it's taken from 1998, when it was approved for erectile dysfunction, to essentially the late 2000s for us to get back to cardiovascular. And indeed, what we're seeing now more and more in basic studies and clinical studies is that it does have important cardiovascular effects.
So there was some considerable doubt as to whether PDE5 was in the heart, had any role in the heart, and your study directly addresses that.
That's right. I mean, PDE5 is expressed, we know, at the gene expression level it's there, but it's very low, and the protein levels are also very low. One of the stories about phosphodiesterase in general is that how much you produce is not necessarily important. It's all about location, location, location. There are very strategic places within the cell that these proteins can reside, and they can control a pool of a cyclic nucleotide that becomes very potent. But still, when you're using a drug like sildenafil or any drug, it's never exactly clear that that's all it's doing. Is it hitting another PDE? Is it hitting it in the myocyte? We know it's expressed in other cell types. Could the cardiac benefits be coming because it's acting as a vasodilator or because it's influencing fibroblasts? All of these things have been posited to explain this. So what we did is essentially create a mouse where only in myocytes we can genetically manipulate the expression of PDE5. And we can do this it's called conditional technique, where if you give the mice a form of tetracycline, doxycycline, then you induce the change in gene expression. So the mice, in the absence of doxycycline, PDE5 in heart muscle cells was very high, much like we see in human heart failure. And the study shows that when that happens, things are worse. We were using a pressure overload model, typical in mouse studies, sort of hypertension, hypertrophy, heart failure model. And if the PDE5 levels were high, it was much worse. Then we could induce the stress. We could produce hypertrophy, produce dysfunction. And instead of giving a drug, we could now give doxycycline back and genetically mimic sildenafil, but only in heart muscle cells. So only in the heart muscle cells now is PDE5 going to be blocked. So this is sildenafil genetically targeted. And it worked. It actually reversed the disease. Very striking. And it not only fixed it in the myocyte, but it also fixes it in the fibroblasts. So fibrosis also gets less. So the myocyte's doing a lot of talking, and this really shows that PDE5 in the muscle cell is important.
It's a beautiful example of going from the clinic to the basic laboratory to document exactly what is happening and how it can be used. Now, this has actually led to a clinical trial.
That's right. Right now, the NIH is sponsoring a multi-center clinical trial called the RELAX trial, which is testing the use of sildenafil. They're using the Revatio, which is the form of sildenafil that's been approved for pulmonary hypertension. That's what the patients are taking. And it's in heart failure with a preserved ejection fraction. So it's that cohort. Typically, those patients have high blood pressure. They have left ventricular hypertrophy. It's a little closer to what was being done in some of the early experimental models, and that may be why we started there first. But there's growing interest in the other form, the dilated, depressed heart failure form of heart failure. And there have been several smaller trials, roughly 40, 50 patient trials, randomized, published in JACC and also in Circulation recently, suggesting that these patients can also benefit from sildenafil. It improves exercise capacity, seems to improve endothelial function and flow-mediated improvement in flow to the skeletal muscle. And there is now talk of a larger, multi-center, more definitive trial of sildenafil in that form of heart failure as well.
Well, this is really fascinating work and very valuable in pointing to potential therapies that we can use in the clinic. Thanks so very much for being with us. For Inside JACC, I'm Tony DeMaria.